Hi, my name is Alexa Chu, and I'll be discussing my research for the New Jersey Space Grant Consortium Academic Year Fellowship. Human spaceflight is a major endeavor and can cause significant changes to one's physiology. NASA's Human Research Program has determined 6 major areas of interest: muscle function, bone health, cardiovascular response, behavioral health, and performance, immunology, and sensory motor function. However, we wanted to ask ourselves, what other areas of study are there? We decided to focus on wound healing responses in a simulated spaceflight. We targeted 2 areas which could potentially alter this, which is microgravity and hypoxia. Microgravity can alter tissue repair through various mechanical chemical factors, and hypoxia through changes in spacesuit and spaceship microenvironments could reduce the oxygen received to the wound. And based on the literature, it suggested that microgravity does impair fibroblast cell migration, as well as hypoxia, which is known to inhibit wound healing due to insufficient oxygen delivery. The goal of our studies was to observe the effects of simulated microgravity in conjunction with hypoxia on fibroblast cell migration. And the way that we do this is typically through something called a scratch assay, in which we plate cells on a 24-well plate, wait until they're confluent, and then induce a wound with a pipette tip. We measure the wound closure over time through imaging the cells. Since we wanted to model microgravity, this is done typically through something called a rotary cell culture system or random positioning machine. These are very expensive, and there are various logistical issues with inducing the wound. So we had to figure out if there's a simpler, less expensive way. We devised something called a flask inversion model for simulating microgravity in vitro, in which cells are seated on a T25 tissue culture flask, and once reaching confluency, we induced scratch with a pipette tip, and we filled the flask with media and flipped them upside down, then placed them in either hypoxia or normoxia. The benefits of this are that this model is inexpensive and it's easier to perform the scratch assay. Here's a figure of how we perform a scratch assay. Like I said, we're performing it in a flask, and this was a 24-hour experiment. Here's a figure. This is what we consider normal condition, typical cell culture position, configuration versus inverted. And here's a figure of the initial wound area across our 4 conditions, combinations of a hypoxia, normoxia, or inverted or normal configuration. So from this 24-hour scratch assay, we saw that at 18 hours, cells in hypoxia exhibited greater percent wound closure. And at 24 hours, hypoxic groups, we say, generally perform the best. We also saw no clear trend between the inverted and normal configuration. And this may seem counterintuitive for the case of hypoxia versus normoxia, because earlier we were saying that insufficient oxygen delivery inhibits wound healing. However, there exist several studies which have similarly found that hypoxia can enhance early skin wound healing. And this is through something called hypoxia-inducible factor 1, which stimulates fibroblast migration through a short-term, this is a short-term response. And for the inverted normal configuration, we suspected that the results are largely affected by the initial wound size, and that since the wounds were too small, the wound area, they were likely to close prior to seeing this full effect of our model. So we concluded that we needed to extend the observation period to observe the full effects of the modeled microgravity. To do that, we increased the initial wound size by, instead of a 1 ml pipette tip, we used a 2 ml aspirating pipette, and this turned this into a 72-hour experiment. Here's a comparison of the initial wound areas. And for the results, we saw at 24 hours, the normoxia hypoxia trend is similar, which is supported by the literature we found. And beyond that, normoxia normal has greater percent closure compared to hypoxia past 24 hours. And we do begin to see a greater difference in inverted versus normoxia. So in conclusion, hypoxia can enhance early cell migration, but it later inhibits wound healing responses. And modeled microgravity does in fact inhibit cell migration. For future directions, we'd like to perform similar experiments using different cell types with this new method. Thank you all for listening, and thanks again.